Hello, folks. I hope you guys are doing well. We have reached a very interesting stage in embryogenesis now, and this is known as the embryonic period or the period of organogenesis. All right, this extends from the third to the eighth week, right? And I've kind of split this into two lectures, and we're not going to really get into the detail of every organ and how it is formed because that is something we shall deal in systemic embryology. But I just want you to know the basics, as in what kind of tissue and how it is derived from which of these germ layers. So technically we'll be speaking about the fate of all the three germ layers that we have seen in the previous lecture, right? So firstly we are going to speak about neurulation, which is a derivative of the ectoderm, rather the neuroectoderm. It is the process of the formation of the neural tube. Then we'll see what exactly happens with the intra-embryonic mesoderm, but we'll mostly be speaking about the paraxial mesoderm, which arranges to form certain cubical structures known as somites. There is also the intermediate mesoderm, which we shall discuss in the systemic embryology of the genitourinary system. And the lateral plate mesoderm we'll talk about only a little when we speak about the intra-embryonic coelom. This shall be discussed while discussing the folding of the embryonic plate. Now there is lateral folding as well as craniocaudal folding, and when that happens we'll also see what is the fate of the endoderm and how it forms what is known as the primitive gut tube, which leads to the formation of the entire gastrointestinal tract. So subsequent details of each of these things will be discussed in organogenesis part 2. Let's first look at the fate of the ectoderm. Now, you've seen what happens at 19 days in the previous lecture. The ectoderm forms a typical structure known as the neural plate. I've also mentioned something known as induction. And induction is essentially something where one of the tissues communicates with the surrounding tissues by a phenomenon known as cellular crosstalk. There's molecular signaling involved. And the notochord is responsible for the induction of the ectoderm that lies over it. That's the notochord, and that's the ectoderm lying over it. And it induces the ectoderm to form what is known as neuroectoderm, which is arranged in the form of a plate like this, which is known as the neural plate. Okay. Now you can also see a groove arising in the median plane, which is known as the neural groove. So now, if you look at a transverse section taken somewhere in this region, it's going to look like this. That's your notochord. And above that, this little U-shaped thing that is happening is actually the folding of the neural plate in the center to give rise to two bulging creases on either side. And these two creases eventually fuse together to form a tubular structure that is known as the neural tube. So the process of formation of the neural tube from the neural ectoderm overlying the notochord is known as neurulation. And while neurulation happens, a couple of cells lying on the crest here are budded off dorsolaterally, as you can see with these arrows, and these are known as the neural crest cells. Now I'm not going to be discussing more about the neural crest cells right now. We'll have a nice little slide on them when we discuss the systemic embryology or the development of the central nervous system. Just know that these are cells that migrate to certain parts of the body, and some embryologists in fact have started calling it the fourth germ layer because the contribution of the neural crest cells is extremely significant. So more on that in another lecture, but let's move on to what exactly happens in the formation of the neural tube. The neural tube does not just form throughout, it starts appearing firstly in what is supposed to be the future cervical region, and it proceeds from a cranial to a caudal direction. So as the neural tube forms from the cranial to the caudal direction, there are certain things that are remaining open for a while, and as you can see, when the folding has completely taken place, you still have the opening on the cephalic side known as the anterior neuropore and an opening on the caudal side known as the posterior neuropore. I just want to tell you a little bit about the primitive streak. By about 21 days, it has already started regressing, and by about 23 days it's almost non-existent. By 26 days it completely disappears. Okay, now the fate of the intra-embryonic mesoderm. Now you already know from the previous lecture that the intra-embryonic mesoderm is classified into three things. Quickly just drawing a basic view, viewed from the amniotic cavity, looking at the ectodermal surface. You have the procaudal plate here, you have the notochord, then you have the primitive node here, you have the regressing primitive streak now, and you have the cloacal membrane. Okay, just imagine that the ectoderm has been removed and now you're looking at the intra-embryonic mesoderm from above, that is from the roof of the amniotic cavity with the ectoderm removed. So what do you see? You see the procaudal plate, yes. You see the notochord, you see the primitive node, you see the regressing primitive streak, and you see the cloacal membrane, correct? Now the intra-embryonic mesoderm arranges itself in this manner. Lying on either side of the notochord you have the paraxial mesoderm, which soon starts differentiating into cubical masses which are known as somites. Adjacent to that you have what is known as the intermediate mesoderm, which gives rise to the genitourinary system, which we shall consider in development of the genitourinary system. And lying lateral to that is what is known as the lateral plate mesoderm. So you have the paraxial mesoderm forming the somites, the intermediate mesoderm forming the genitourinary system, and the lateral plate mesoderm. In the lateral plate mesoderm start arising small cavities, and these coalesce to form a bigger cavity which is now known as the intra-embryonic coelom. Okay, so if we take a transverse section somewhere over here, then we are directed to what you see in the diagram here. You can see the notochord, you can see the red colored intra-embryonic mesoderm on either side. That's the amniotic cavity. That's the yolk sac. Don't even worry about the yolk sac and the endoderm right now. Okay, so you see these little bulges occurring. That's essentially the paraxial mesoderm. Lateral to that you have the intermediate mesoderm, and lateral to that you have the lateral plate mesoderm, and you are seeing intercellular cavities appearing in it. Okay, now this is somewhere around day 19. Let's go to day 20. At day 20 you can see that the neural groove is formed. The paraxial mesoderm is getting more and more thicker, and eventually by about day 21 you have the emergence of somites. At the same time, all the little cavities in the lateral plate mesoderm coalesce together to form a space that is known as the intra-embryonic coelom, also known as the intra-embryonic body cavity. This is going to give rise to the future pericardial, pleural and peritoneal cavities. More on that later. Okay, by day 21 you can see that the neural tube is completely formed and the somites are lying on either side of the notochord, above which lies the neural tube. So you can see the notochord here. Okay, I hope this is clear. Now we are going to speak a little bit more about the paraxial mesoderm, to be precise, the somites. I just want you to observe something though. Can you see that the amniotic cavity is slightly folding in a transverse section, which means that the lateral ends of the amniotic cavity are coming close to each other and the amniotic cavity is becoming bigger. Now this is the advent of what is known as the lateral folding, which we shall study later. Let's talk first a little bit about the somites. Now the somites are a very interesting thing. The embryologists love it because counting the number of somites gives them an approximate idea of the age in days of the embryo. So by day 20 the somites start arising. About each day 3 new somites are added till the end of the 5th week. By the end of the 5th week you have about 42 to 44 somites. Okay, and what are these? These are cubical masses which are nothing but an evolution of the paraxial mesoderm. I want you to know, however, that there are certain things lying just above the somites, and they are exactly like the somites, but they are known as somitomeres. Okay, we're going to discuss more about that later too. These can further be divided into occipital somites and pre-occipital somites. Okay, so we've got the somitomeres and the somites all arranging themselves laterally on either side of the notochord and the neural tube. Correct? Let's see how it looks in a transverse section. This red colored donut here is a somite. Soon it starts differentiating into a ventrolateral region that is known as the sclerotome, and a dorsolateral region that is together known as the dermatomyotome. Okay, let's go to diagram number C. Here you can see the gray colored dermatome, intermediately the golden colored myotome, and the light blue colored sclerotome. Now I'm sure you already know what's happening here. The dermatome will give rise to the skin of the body wall, the myotome will give rise to the muscles of the body wall, and the sclerotome will give rise to the bony and the cartilaginous structures. Right? Now this black thing over here is the notochord. This blue thing over here is the neural tube. And this is the loosely arranged mesenchyme that is going to contribute to the axial skeleton. Inside it is going to be the remnants of the notochord, and everyone knows what that is called. That's known as nucleus pulposus. This little golden thing is going to give rise to the muscles of the body wall, while this thing over here is going to give rise to the skin of the body wall. Okay? So I want you to take a minute and understand how this is going to be converted into what would look like a transverse section of the body wall. Okay, so you'd probably be having nucleus pulposus inside the body of the vertebra. Then you'd be having the entire vertebra formed, something that would look like this, right? And this is all formed by the sclerotome, and inside you have the vertebral canal, and inside the vertebral canal you'll be having the spinal cord. Now technically this neural tube at this region will form the transverse section of the spinal cord, while as it ascends upwards it will also form the brainstem and the brain, but that is not our topic for today. I just hope you understand how this notochord remains as a nucleus pulposus inside the body of the vertebra, and this entire sclerotome properly forms the entire vertebra at this level, and the neural tube thus embedded inside forms the spinal cord. I want you to use your imagination here, and let's move on to the next slide. So now we are going to be discussing something that is known as the folding of the embryo. As I mentioned before, the folding is occurring in the craniocaudal direction, which can be called as longitudinal folding, and lateral folding, which can be best observed by seeing a transverse section, right? So let's first talk about the craniocaudal folding. Okay, I'm once again going to draw the same embryonic disc that I drew before, the slipper shaped structure, procaudal plate, notochord, primitive node, primitive pit, a primitive streak that is on its way to disappear, and the cloacal membrane. Now I've already discussed before that the procaudal plate and the cloacal membrane are the regions where there is no intra-embryonic mesoderm. The ectoderm is in intimate contact with the endoderm, correct? But everywhere else there is going to be the intra-embryonic mesoderm. We've also established that on either side of the notochord you've got the paraxial mesoderm forming the somites. Then you've got an area known as the intermediate mesoderm giving rise to the genitourinary system. And you finally have the lateral plate mesoderm that is going to have some cavities inside it. And these cavities are essentially going to coalesce to form a bigger cavity that is known as the intra-embryonic coelom. If I were to draw the intra-embryonic coelom, it extends as a horseshoe-shaped cavity on either side of the notochord in the lateral plate mesoderm, and this horseshoe meets cranial to the procaudal plate. Okay, this horseshoe shaped cavity meets cranial to the procaudal plate, and on either side this horseshoe shaped cavity is in contact with the extra embryonic coelom. So what I drew is the intra-embryonic coelom, and these are the two regions where it is in contact with the extra embryonic coelom. Okay, now just understand this. The mesoderm lying cranial to this union or this horseshoe shape part is known as the septum transversum. Okay, this is known as the septum transversum. It is going to give rise to the future liver and diaphragm. Okay, this region of the intraembryonic coelom is actually going to give rise to the pericardial cavity. Okay, don't worry about how, we're going to speak about all that in the development of the heart tubes. But this is the pericardial cavity, and in the floor of this cavity is a mesoderm that is known as the cardiogenic area. Okay, so if you were to now describe the slipper shaped embryonic plate as viewed from the amniotic cavity roof after the ectoderm is removed, you see the following structures from cranial to caudal. Number 1, septum transversum. Number 2, the region of the intraembryonic coelom that will form the future pericardial cavity, in the floor of which lies the intraembryonic mesoderm that is known as the cardiogenic area. Number 4, the notochord and the somites lying on either side. Number 5, the primitive pit and the regressing primitive streak. And finally, number 6, the cloacal membrane. So once again, from cranial to caudal, you have the septum transversum, the intraembryonic coelom that will form the future pericardial cavity, in the floor of which lies the cardiogenic area, then the notochord along with the somites lying on either side, then the primitive node and the regressing primitive streak, and most caudal, the cloacal membrane. And now let us discuss what happens when this starts folding. So how is it folding? Let's have a look at this diagram. It's folding in this way. This is the cranial end, this is the caudal end. Okay, what you see in blue is the neuroectoderm. You can see it's already bulging in this area because that part of the neural tube is going to form bulkier structures like the forebrain, midbrain, correct? Then you can see that the amniotic cavity is increasing in size and the yolk sac is reducing in size, correct? Okay, let's go further to diagram number C. Now we are somewhere at 24 days. Okay, look what's happened. The neural tube is really bulged in this area. The amniotic cavity is becoming even bigger. The cranial and the caudal ends have come really close to each other. And see what's happening to the yolk sac. The yolk sac is nice and round here, but inside over here, the yolk sac is now forming a tubular structure, and that tubular structure, my friends, is known as the primitive gut tube. Okay, that's the primitive gut tube. So this little connection that lies between the primitive gut tube and the yolk sac, which is still keeping both the ends connected, is known as the Vitello-intestinal Duct. Okay, so now you've got the primitive gut tube, and somewhere in the center you have the Vitello-intestinal Duct connecting it with the yolk sac. Okay, at the same time the amnion is becoming much bigger, the cephalic and the caudal ends have come much closer, the neuroectoderm in the cranial region has become much thicker, and the red colored mesoderm has already started showing a few things. Remember, the septum transversum it's now going to come and lie over here. This was the cranial most structure. Remember the cranial most structure, but now after folding it is coming to lie over here somewhere in the future chest region. Above that is your pericardial sac with developing heart tubes. Okay, don't worry, we're going to visit this diagram again. Just understand that what was lying caudal to the septum transversum is now going to lie cranial to it, and the floor that was supposed to be the cardiogenic area has now become the roof, and that roof forms what are known as the heart tubes, which we shall study in the development of the CVS. So now you've got a heart tube underneath which lies the future pericardial sac. Okay, great. Also, the allantoic diverticulum, which was connecting to the connecting stalk, seems to have shifted somewhere in the center, just below the chest region. Let's just call it the future umbilical area, right? That's your allantoic stalk, and that is your connecting stalk. And both of these are going to be now devoted towards the future umbilical area. Okay, don't worry too much about this. We're going to revisit all these things in systemic embryology. So what we just studied was the longitudinal or the cephalocaudal folding. Now let's move ahead to how the lateral folding occurs. Before we even start that, I want to tell you what exactly happens because of the arising of the intra-embryonic coelom. It now divides the lateral plate mesoderm into the area that is in touch with the yolk sac, that is known as the splanchno-pleuric or the visceral mesoderm, and the mesoderm that is in contact with the amniotic cavity and the surface ectoderm, which is going to form what is known as the somatic or somatopleuric, or you can also call it the parietal mesoderm. Okay, so this is the lateral folding. You can see it happening, correct? By about 24 days, the neural tube is already formed. So whatever remains of the ectoderm is known as the surface ectoderm. And the surface ectoderm on either side eventually, by the end of the 4th week, literally connects to each other, and thus the intra-embryonic coelom on either side now becomes a continuous cavity. The intra-embryonic coelom on either side becomes a continuous cavity that is going to form the future peritoneum, and the part that was in contact with the yolk sac, as you can see, is now only going to remain in contact with the primitive gut tube, that is known as the visceral mesoderm, and the part in contact with the surface ectoderm is going to be called the parietal mesoderm, correct? So this bubble was pinched off, and there is only a connection between both of them somewhere in the future umbilical area through a Vitello-intestinal duct. That should give you an idea that the Vitello-intestinal duct remnants will be one of the contents of the umbilical cord. Anyways, for those who don't get that now, you will realize everything as time passes. Don't be scared and don't be anxious about this. It's really easy once you start applying yourselves more. So you can see the amniotic cavity is completely surrounding the surface ectoderm. The neural tube is going to form the future central nervous system. The notochord is almost going to regress and remain as the nucleus pulposus, and the future body cavity is already formed, and inside it is wrapped your primitive gut tube, which is surrounded by the visceral mesoderm, and the surface ectoderm that meets each other is completely lined by what is known as the parietal mesoderm. So that is a brief review on the folding in the lateral aspect, where it comes to meet each other in the midline. Now there is a plethora of clinical syndromes that occurs due to abnormal folding, omphalocele, etc., etc. I really do not want to discuss that in general embryology. We have to discuss these things as we discuss the systemic embryology. So let's go ahead and have a quick revision of part 1 of organogenesis. At 19 days, you can see the typical neural plate that is formed, the notochord, the primitive node, the regressing primitive streak. Okay, by 20 days, see how somites have started arising in the paraxial mesoderm. Also, the neural groove is formed and nicely deep. Now, you can see that the neural tube has already started forming in the future cervical region, and the number of somites are increasing. They are added from the cranial to the caudal direction. The length of the embryo is increasing, and you can see the anterior and the posterior neuropore still open. We are at the age of about 22 days. By about 23 days, this is a general picture of what it looks like. Let's go ahead and look at the transverse section. Once the neural tube is completely formed, whatever remains out is the surface ectoderm. It is going to contribute to the epidermis of the skin. Please remember, while the dermis of the skin is going to be contributed by the dermatome aspect of the somite. Now you can see a couple of neural crest cells that are going to migrate on either side, whose fate shall be discussed in another lecture. You can see the notochord here, and if you look at it from the ectodermal side, you can see the notochord here, and on either side you can see the somites that are being formed. Beautiful pictures from Langman's. This is a picture of an approximately 28-day embryo. You can see how the yolk sac is out, and there's a small connection somewhere in the future umbilical area, which is known as the Vitello-intestinal Duct, and this is the cranial end and this is the caudal end of the developing embryo. So by the end of 28 days, or shall we just say by the end of about 4 weeks, this is what the embryo looks like. This is the head end. You can already see the arrival of a placode that is known as the lens placode. There's also the arrival of the otic placode, future ear area. Don't worry about that. Then we've got this entire surface ectoderm underneath what are lying all your somites. Okay, more about that in the next lecture. This area is known as the pericardial bulge, where the future heart is going to develop, under which is going to come your diaphragm and your liver. I hope you're kind of connecting the dots here. Remember how the heart lies over the central tendon of the diaphragm? So that's going to be your central tendon of the diaphragm. That's going to be your heart. Yeah? And there's a certain thing that appears between the head bulge and the pericardial bulge, and these are known as the pharyngeal arches. Incredible structures having contributions from all three germ layers. More on that in a separate lecture. So somewhere here is going to be your Vitello-intestinal duct connecting with the yolk sac. Okay, things are getting seriously beautiful now, so I hope you're having a great time. I'm going to see you in the next lecture, Organogenesis II. We're going to discuss a little more about every germ layer and what they contribute to the formation of. And then we're going to end the series with a nice lecture on the placenta and its evolution from that second week cytotrophoblast stage to a beautifully formed, well-organized organ that is responsible for supplying nutrition to the fetus. Thank you so much for listening. See you in the next one.